The research was conducted by Philip Zimbardo, and then the prison was constructed by a few of his peers. And then the police cooperation, which is I think something that's really interesting, is that the local police department, as well as other police departments, got involved, and then Stanford University security. So, the purpose of the study is Zimbardo and his colleagues are interested in finding out whether police brutality reported amongst guards in American prisons were due to the sadistic personalities of the guards or the prison environment. And this is actually something that he later talked about in his book, The Lucifer Effect, which I have mentioned later. But he kind of wants to see if it's just like the environment that people are in which causes them to be bad, or it's just like them within themselves. So he predicted that it was the situation, the environment that people were in, that made them act the way they did rather than their personalities. Okay, that's not how we do it. So the procedure was he advertised on Stanford campus for students to participate in a psychological study that affects prison life, and then he advertised it, paid him $15 a day. So he got 75 applicants, and then through that he found 24 men who he deemed to be, kind of in the middle. So they weren't like too crazy; they were just like pretty average people. And then the participants were randomly assigned to either play the role of a prisoner or the guard. So there were a total of 10 prisoners and 11 guards. And then the setup, this is the part that I find really interesting. So he booked the prisoners like actual prisoners. Like he just pulled up at their house and was like, yeah, you guys are arrested. And this is one of the ethical concerns that is brought up with the issue, is that nobody was really disclosed on the information that would happen. They were just arrested. They were fingerprinted and booked and treated like actual criminals. And then once they were at the police station, they were blindfolded and then taken to Stanford University in the basement of one of the department buildings where Zimbardo had created his prison. And so he began his de-individualization process by stripping them down, taking away all their possessions. He put like a little anklet collar on them. And then he gave them prison clothes, a tight hat, like a tight little like latex hat thing. And then he put like numbers on them and then threw them in a cell. And so this is just kind of like that process that they went through. And this is the guards. The guards were also, they had their own process where they were all given the same uniform, given dark glasses, and kind of like debriefed on what they had to do. Oh yes, this is the part that I just mentioned. So the guards were all dressed in identical clothing to take away their identity. So, they kind of like lacked that responsibility of that individualism. They were given a billy club, which was actually donated by local police departments. They wore sunglasses to make eye contact difficult. And then the prisoners, like I mentioned, were also dressed identical. And then this is something that I found really interesting too, is that the guards and the prisoners had kind of fallen into the role. So the prisoners began to act like prisoners, and then the guards began to just act like guards. And so this is kind of that process that I was talking about, where they were blindfolded, and then you can kind of see like, it looks kind of janky, like that's actual wood, but that's the prison that they constructed. And so the guards were given full control and power, and this is something that Zimbardo had done in terms of his research method, in the sense that he just gave the guards full authority and power. And what he did was he was more of a observational researcher. So he just kind of like watched them do what they wanted to do. And so on the first night, the guards were given the authority and power to conduct their counts, which is when they would take the prisoners out. And this is kind of like that de-individualization process that he was talking about. He would just make them count off their numbers. So they were just like woken up, and they just had to like say their numbers. And then the guards were told that Zimbardo was the prison superintendent, so he was to be reported to. So this is kind of something that he talks about later, in which they kind of lost that, what do you call it? Hold on, give me 1 second. Kind of like lack that responsibility, because they're like, oh, the prison superintendent told me to do it. And so punishments is something that the guards did practicing. So they taunted the prisoners, they gave them petty orders. It was pretty pointless. And so the most common form of this was push-ups and sit-ups. So they would just like make the prisoners do push-ups and be like, yeah, bro, just gotta do push-ups. And then there's a certain threshold that people can take. So then the prisoners began to rebel too. And this is interesting. The guards actually used fire extinguishers, and they shot like carbon dioxide at them because it was so cold. The prisoners just like back away back into their cells. And then they also stripped them naked and took away their beds. So at this point, it's like day 6 in the experiment. The guards are absolutely crazy. They're just wilding out with power, and the prisoners, they begin to rebel a little bit. That's something that, just to kind of keep it concise, I didn't mention. So around hour 36 of the experiment, this prisoner number 8612, they began to suffer from acute emotional distress, rage, uncontrollable crying, and disorganized thinking. And so this is really interesting because they really had fallen into that prisoner role, and the guards really began to break down these prisoners, which is not something that Zimbardo instructed them doing. It's just kind of like they fell into that role of it. And then prisoner 819, the priest actually had been called in to kind of like help de-stress the prisoners. So he began breakdown crying hysterically, and then he was taken into another room where Zimbardo and a few of his peers began to talk to him. So his chain was taken off, and all his clothes were given back to him, and he was just taken into another room, given some food, and taken to see a doctor because the stress of the experiment had finally got to him. So the experiment was supposed to last 2 weeks, but it only lasted 6 days and was shut down early because of the amount of emotional breakdowns that occurred from the prisoners, and then acts of excessive aggression from the guards. And so the end conclusion of all this is that Zimbardo concluded that when people are put in situations where they feel anonymous, that's kind of what I was talking about, they just use excessive power over others and begin to depersonalize others and become evil because they feel like it's not them that's doing it, and they can kind of push it against the higher power. He also discussed how people conform to the social roles when they are expected to, and this is kind of something that you see early in the experiment, where the guards begin to act like guards, and the prisoners began to kind of conform into this prisoner identity. So ethical issues with the experiment is that Zimbardo does not disclose to the participants what was going to occur. So he really didn't tell anybody what was going on. He didn't tell the guards they were going to act like guards, and he didn't tell the prisoners that they were going to be prisoners. The prisoners did not consent to their arrest. The prisoners were not expected from psychological harms of the experiment. And we do see this with prisoner breakdowns. And then these are my works cited page. And further reasoning, you guys can read about the Milgram experiment, the Lucifer effect, that's the book that I mentioned earlier. The American Psychological Association is great for reading these research. And, you guys can definitely defund the police if you want to. So I just have a quick link right here. And yeah. Okay. Thank you.
I had a question. I did some research on the Stanford prison experiment after reading your article, and one of the things I saw was that apparently Zimbardo did actually have like some one-on-ones with guards during the experiment, where he encouraged them to be like tougher and put more pressure on prisoners and act more guard-like. And apparently some of the guards even admitted that when they were told that, they stopped thinking like real like prison guards and started thinking like movie roles. Like they said that they wanted to fit that evil guard. Do you think that had a pretty significant impact on the outcome of the experiment, or do you think that that's like a natural result of being in that environment, that that environment is conducive to playing these movie caricatures of the bad
guy? So I really think it just kind of breaks down into this, I guess, like that stigma that people associate. So when he said guard, these are just college students that really had no idea what actual prison guard was like. So I do think that you are right in the sense that Zimbardo having these one-on-ones with these guards did play a solid effect on how they acted and responded. Are there any other questions?
I also had a question. So, I mean, I learned about the Stanford experiment in AP Psych, so my senior year of high school, and like we also learned that now a lot of new evidence is coming out that like the Stanford experiment is getting like debunked, as well as like a lot of earlier like psychological experiments or experiments that happen in psychology, because like they were done unethically. And I think the Stanford experiment is like kind of like what a lot of people use as an example, because a lot of the things they didn't really like. Apparently like the prisoners were told that they couldn't leave, even though in the beginning they were told like, oh, if like you want to leave you can, but then later in the experiment they were like, no, you can't leave even if you want to. So I think, do you think that they should like conduct more experiments about stuff like this, like role playing and stuff like that now, to see if like anything has changed now that there's like more evidence debunking the experiment? Like, what do you think they could do to change the experiment if they did it now?
So I think the experiment was really important because it kind of offered like what not to do in terms of the realms of experiments. So it was kind of one of those crazy things. It's just like, we're like, yeah, we're not going to do that again. But I definitely do think that if we are able to conduct it in more of a, ethical manner, then it is something that we should see. And in his book that he mentions, I actually had an opportunity to read it, The Lucifer Effect. He kind of does talk about how people in positions of power do abuse their roles. And this is something that you see with U.S. soldiers going overseas, in the sense that they lack that self-identity and that responsibility. And, I kind of did mention the debunk the police, or sorry, not debunk, defund the police, because, you do see a big abuse of power when it does come to police officers. So, but I think if we are able to conduct the research again in more of a thought out, well put manner, then it is something that we should see.
Do you think the sample size also played a role in how the experiment carried out? Because as I understand it, they were all young white males that were included in the experiment. Do you think having a more diverse group would lead to like a different outcome, or like a more extreme outcome, less extreme outcome?
I think, the race of the participants did play a big role and outcome on it. I think if it was more diverse, we would see, I think we would see more resistance in terms of the prisoners. And yeah, I think that would make things interesting. Yeah.
One thing I wanted to say, going off what Rabia said, was that I was reading my psych textbook the other day, and it was like talking about de-individuation and stuff like that. And one of the experiments it talked about, I can't remember who conducted it, but it was just talking about like the general setup was like you have one person on one side of the room, and they're giving the other person on the other side, who's like an actor who already has a script, of like they're not actually receiving an electric shock, but the person in the experiment, they're like increasing the level of the shock from like 45 to 60 to like 450, but the other person receiving the shock, they like actually aren't getting it, and they have a script saying like, oh my god, please stop, like this is hurting me, and they're just like supposed to scream and stuff like that. And I think it might be Milgram, but I'm not sure. But it was said that even the authority figure, they come in and they say, keep on giving the shock, like you have to give the shock. And they found that even repeating that experiment, over 60% of participants, like they'll still give that shock anyway. So maybe it just like goes to our like socialization, like how we're all taught to like always listen to like an authority figure, like a teacher or someone who has power. Like we're all conditioned, I feel like, to listen to them. So maybe that kind of plays a role in it. I don't know, but like what Rabia said kind of just reminded me of that.
Yeah, and people in the chat are saying that is the Milgram experiment. But I think it is interesting that you mentioned that. So
I also think that that experiment that you talked about is so relevant in the times we're living like right now, because it kind of like what we see and like what you can see on the media showing like how like like I've seen that like the police are taking like the knee one day, but then the next day they're continuing to like tear gas and things like that. So like they're continuing to do their roles even though they might not agree with like what they're doing, which is kind of like what the Stanford experiment is showing. Like people are, it's not the same obviously, but like people are continuing to be in like the roles of authority and like following what they've always been told, even if they don't fully agree with what they're doing.
Yeah.
Does anyone else have any questions? If you don't want to talk, you can always just say it in the chat as well.
Hey, I don't know if I'm fully correct about this, but I remember learning about this earlier. Like I was looking through some research journals, and I think like in the study, like this study was proven to like be not fully accurate because the people in the experiment were told that none of this was real before it was started. So like they knew, so like to them the recording was like they knew that the sound was pre-recorded, and they also knew that nobody was in danger. So like even like the people who stopped, they stopped because they wanted to, but they had previously been told that nobody was going to get hurt and everything was just like a simulation. So I think that might change some of the experiment, like the viability of it, but I do think that a lot of the lessons still hold.
Could you do these experiments today?
Personally, I mean, I wouldn't because I don't.
So what's the problem with doing these today? There's one substantial stumbling block to doing these types of experiments. Do you know what it is?
Yeah. So the main, it's if
You've
I
already
I
known
mean,
about that.
I'm assuming it's the ethical issues. That,
Yeah. So there's
occurred with
something
the-
you guys know about the IRB, Institutional Review Board.
Yeah.
So
Mhm.
they would absolutely not allow this to happen, right? Because the problem is that these caused psychological harm, even to the participants, right? I mean, not even. That's like the person on the button end of the shock. That's quite disturbing to be doing that. The other reason I think it wouldn't work is because I would hope that you guys would know that that NC State would not allow Dr. Guller or myself to shock a student, right? And therefore, if I told one of you that you're going to deliver a shock to another student, I think you should just push the button because it's obviously BS, right? So, so the ethics have changed in such a way that it almost undermines the ability to do the research in the first place. Okay, let's try to do it, Dr. Dollar.
Dr. Rose, do you think that since the ethics for carrying out such an experiment have changed in like a controlled laboratory environment, do you think the like social analysis of real life events where results like this experiment's results are occurring is more important now because you can't replicate this in a controlled environment, without breaking ethical limitations?
Yeah, so that's a really interesting point. So you have like sociological observations that you can make, and you can do analysis of like real worlds. Like you can't do these exact experiments, but there are other types of experiments that you can do to get at issues of empathy and sort of obedience. So like there's another line of research where people have found like that. So there's like I can't remember the exact experiment, but they have a robot that it's clearly a robot. It looks like, R2-D2 or something like that. And you're told to like put it in the closet, and then the robot starts saying like, I'm sad, let me out, please, And so obviously like the participant knows that it's like not an animate object, but yet those experiments, elicit like sort of an emotional response. So you can do things like that. But yeah, it's challenging. I mean, we're not allowed to abuse students anymore, so we kind of missed that train.
So up next, we have Zainab talking about metabolism and how HNMR spec can be used to study that in thyroid cancer.
Okay, so I'm just gonna share my screen. Hold on. Can you guys see my screen?
Yep.
Okay. Okay. So, hi, I'm Zainab, and I read a research paper about metabolic reprogramming detected by HNMR spectroscopy in human thyroid cancer tissues. So, just like a quick overview of what the research is about. So, thyroid cancer cells are known to demonstrate an increase in oxidative stress and decreased antioxidant action, but its effects on cell function are unknown. So that was basically the point of the research, to figure out what changes increased oxidative stress has on the metabolism of cancer cells, and they study this using NMR spectroscopy. So just like a quick rundown of all the basic terms and stuff. Oxidative stress is when healthy cells are attacked by free radicals, and it can lead to cell damage and is associated with cancer. And again, the downstream effects of it on biochemical pathways are unknown, so that's what they're trying to study. And they know from previous research that increased oxidative stress is present in the blood of patients with thyroid diseases, and the reduced expression of antioxidant seleno enzymes, glutathione peroxidase, and thyrodoxine reductase in thyroid cancer. And the main function of these antioxidant enzymes is to reduce H2O2 to water and catalyze reduction of proteins. So given that, they created their method, and it involved patients with thyroid diseases like thyroid cancer and hypothyroidism, stuff like that. And they performed thyroidectomies on them, which is basically removing parts of the thyroid gland. And they took 11 tumor and 10 healthy tissue samples, and then they performed the spectroscopy on it. So this is like a way more like detailed procedure, but I just wanted to highlight like the main steps of it. So first they had their frozen tissue samples and they homogenized it with an ethanolic solution 3 times for 30 seconds to maintain temperatures at different intervals. And then they used an ultrasonic disintegrator on them at 20 kilohertz. And the spectroscopy analysis was done using a 60-degree flip angle pulse at 40-second intervals. So they did this to analyze, again, the differences between the healthy and tumor tissues to clarify whatever changes occurred in the metabolic pathways in response to increased oxidative stress. So these are their results. First, they looked at like the overall metabolites in the cancer in healthy biopsies. And there's the L-phenylalanine and lactate. They had positive loadings on latent component 1. This basically means there were higher concentrations of them in the cancer tissue, and that's highlighted by the red parts here and here. And these metabolites had contributed more than 80% of their variance to latent components. On the other hand, we had myoinositol, citrate, and threonine. There were higher concentrations of these in healthy tissue, meaning there was less of them in the cancer tissue, and they're represented by green. They also looked at specific biochemical pathways. So the first one was the glucose and pyruvate metabolism. They noticed a significant increase in lactate and insignificant glucose discrepancy in the cancer tissues. Additionally, an increase of alanine and acetic acid in the pyruvate metabolism, and an overall increased glycolytic activity in the thyroid cancer tissues. They also studied amino acid metabolism. They noticed significant increases in tyrosine and phenylalanine, and these are basically called aromatic amino acids. And they also noticed an increase in branched chain amino acid isoleucine right there. They also studied lipid metabolisms, and overall there was insignificant differences except for increased myoinositol in healthy tissues and decreased citric acid in cancer tissues. And lastly, they studied energy and redox balances. Again, insignificant differences in this, but there was a significant increase in formic acid and also in the pool of glutathione. So what all of this basically signifies is that cancer cells tend to have increased production of free radicals alongside a weakened antioxidant defense system that can't fight the free radicals, which leads to an increase in oxidative stress, impairing cell growth and mitosis. So this signals like the imbalanced antioxidant systems in the cancer cells, and the decrease in glutathione peroxidase and thyroidoxin reductase enzymes suggests that the antioxidant systems can't counteract the effect of free radicals, either due to a deficit of those systems or increased free radicals. So the NMR spectroscopy analyses show metabolic reprogramming in thyroid cancer tissues. The decreased citrate and increased acetate levels in cancer tissues support lipid biogenesis, which is basically the synthesis and degradation of lipids involving like the storage for fat for energy. We know that mitochondria regulate homeostasis and energy production processes, so this can dramatically increase or decrease based on altered activity, leading to enhanced glycolysis. Their study findings show increased glycolytic activity in thyroid cancer cell metabolism confirmed by the increased lactate concentrations. They also suggest that lactate may promote oxidative stress resistance due to increased lactate and choline levels alongside lower levels of citrate, glutamine, and glutamate. I'll answer all the questions like later. Okay. So, where was I? Their study findings show increased glycolytic activity, and they also suggest that lactate may promote oxidative stress resistance, said that already, and the elevated lactate levels result from increase in the glycolytic pathways in cancer tissues, and their studies show that it is involved in altering lipid profiles and fat concentrations alongside high choline levels in cancer tissues. their big finding and result from like the study is that there's a correlation between oxidative stress and the metabolic profile in thyroid tumors, which may be due to the reduced activity of the Krebs cycle. NMR spectroscopy results show reduced citrate, which is produced during the Krebs cycle, and this may be attributed to a decrease in oxidative metabolism consistent with an increase in acetate. The studies, okay, hold on. So let me just talk about this diagram first. So the diagram basically shows altered metabolism in thyroid cancer cell. There's reduced citrate, but an accumulation of tyrosine, phenylalanine, isoleucine, etc. And again, this can be attributable to a decreased Krebs cycle activity, suggesting a shift towards glycolysis with changes in the metabolites involved in mitochondrial activity and in the glucose pyruvate metabolism. So the focal finding here is that thyroid cancer cells can reprogram their metabolic activity to survive in conditions of high oxidative stress with a compromised antioxidant system. And thyroid cancer cells have metabolic plasticity that allows them to survive in extreme environmental conditions because they're able to reprogram their metabolic activity in order to respond to all that increased oxidative stress. That is it for my presentation, and I'm going to try to see what all these questions are. Wait, hold on. Okay. Did everyone see that? Ugh. Okay.
I think those questions are about the last presentation. We'll probably do that after.
I was like low-key freaking out that everyone's asking all these questions.
No, you're good. You're good. I guess for me, you were saying that these cancer cells can survive in like these high stress situations. Do you think it's more so that like the cancer cells begin, like they form because of the high stress situations, or they're just able to survive them?
So that's what they, that part is still kind of unknown, but they do hypothesize that since like, you know, cancer cells, they're malignant, they can like multiply and essentially they don't die. So they think that being in like increased stress may have induced them to be cancerous, but then instead of dying from it, they adapted to it, while the healthy cells or like cells that couldn't attack them died instead.
Okay.
Yeah.
And I don't know if you've read up on any of this, but like, do you think this applies to like other types of cancer other than thyroid?
That is interesting. They didn't specify if it applies, but I assume it would. But again, I'm not a scientist. But it is interesting, like in just like one part of the body they have cancer cells that can adapt to this. So it would be interesting to see like similar research that shows like you know, lung cancer or anything like that, if it responds in the same way.
Going off what Shabazz just said, I think from my biochem class, we talked about how cancer cells just in general, they've seen like the trend of them like getting their energy from glycolysis. I don't know if any research that specifically talks about like oxidative stress like this one does, but I think like the general trend is just that they do like have increased glycolysis. And I guess my question was like, based on the amino acid figures you showed us, I wonder why it's only like, I know that tyrosine and phenylalanine, like those those aromatic ones, they feed into the Krebs cycle. Mm-hmm. But from what I remember, there's like I think almost every amino acid except leucine and lysine are glucogenic, so they do feed into like that whole chain. So I wonder why there isn't like a significant increase in them. I don't know if my question makes sense, but
I get that. Yeah,
it's interesting, like the trends, like in which amino acids are like used up and which ones aren't.
Yeah, they're just they try to say that by saying that something's wrong with the Krebs cycle activity, causing like some of them to be increased, but others to like the intermediates to not work. Like downstream, you know, like the cascade effect, causing other metabolites to not work and other cycles to not produce as many.
Okay.
Any other questions?
So I don't really know much about like cancer cells or like oxidative stress, but I was just wondering like, is this basically saying that or thyroid cancer cells grow better under oxidative stress than if like there was no stress?
Not necessarily. It's saying that when they are in that oxidative stress, they can adapt better to it and survive. But it doesn't like necessarily mean that, you know, like from point A to get to point B, they need the increased oxidative stress for them to like become cancerous and stuff.
So do normal cells like they have no effect whether like the stress level is high or low?
They do. They they talked about like the antioxidant system. So like when there's an accumulation of the free radicals that attacked the healthy cells, some of them could not counteract the free radicals due to a weakened response. So that's why some of the healthy cells, like, you know, they basically died or became cancer cells, or they adapted because their antioxidant response system was better. So some of the healthy cells could and some couldn't. But they are affected by the oxidative stress, yeah.
Yeah, Rabia, I think in general, just when you have low oxygen, it's really bad for you. Because I remember in biochem, we learned how like hypoxia, so like having low oxygen when you're an infant or like when you're really young, it's super bad for you. And I think it can lead to blindness and stuff, just because like that free radical is like so disruptive to like the electron transport chain and stuff like that. Like it will literally kill your healthy cells. So.
Oh, okay, that makes sense.
Yeah, I probably should have said oxygen.
Do you think this research into how free radicals affect cancer cells will affect like treatment going into the future? Because I know like some chemotherapy, as I understand it, at least, can lead to the formation of free radicals.
Mm-hmm.
This will like change how we approach cancer research to try and eliminate states where cancer cells are more likely to survive than non-cancerous cells.
Yeah, that's a good question. I think if more research and like there was more of a broad audience for it, I think it definitely could lead to research or like trying to find some kind of cure. But I feel like this study, I couldn't really find more about this type of research anyway. So I feel like this topic right now is like in its preliminary stages. So hopefully over the years it grows and there's like a bigger audience or more research based on it, that eventually they try to find treatments or therapies like associated with oxidative stress.
So John, to kind of answer your question, so like for everyone who read John's question who doesn't know about the Bandura clown experiment, I believe that experiment was done to like figure out more about observational learning. So I think that one experiment had like a group of kids watching this doll get beat up by an adult, and then when they're alone, they were measuring like if the kids also hit the doll. And then they found that the kids who did see like a violent person interact with that doll, they were more likely to also act violent towards the doll, versus people who didn't see like that violence towards the doll, they were also not likely to act violent towards the doll. And I'm not sure if it's ethical, because my psych textbook always says that it's very controversial, but it never goes into details about why it's controversial. I guess just because it's like showing violence to children maybe. But I think that knowledge is definitely worth having, just like understanding like how we learn, like, and just like how like observational learning works, because I think it's like just so important. But that's just my opinion about it. So
yeah, from what I know about it, some of the kids later went on to be more aggressive than I guess what average aggression would be. And I don't, who knows if that was just them to begin with, but like that's one of the concerns.
So I think that the Bandura experiments would be fully supported today with IRB approval. You can do things that are stressful, and you know, if I think it would be very hard to find a correlation between that experiment and like future behavior in the kids. That is, you know, you get into an issue that we refer to as dosing. So like, you know, is one experiment or even a small series of experiments going to like change the life, you know, the life trajectory of those kids, especially relative to our society is pretty violent. And if you know, I'm watching television, my kids are seeing cops kneeling on people, you know, and so it's really hard to correlate that with like a specific line of experimentation. But I mean, I think that the general question that you guys are asking is what is the acceptability of doing things that are potentially harmful for the sake of science? And the answer is unequivocally that we do a lot of important science that is potentially harmful. Every single drug that we develop is potentially harmful. And, that so you guys are having really interesting, like you're pulling up sort of the key psychology studies about this, but I think an even more salient conversation to be had would be about like drug development. There's such a hard push today for developing treatments or vaccines for the coronavirus. And, we're cutting corners on that. And there's ethical concerns about cutting corners. There's ethical concerns about not cutting corners, and there's a massive misunderstanding about the process, right? So some people think that they've been denigrating like blind studies, the importance of placebo studies, right? So a lot of the, like, especially the conservative media is talking down about, that, saying that it's unethical to have placebo groups because, everybody should be given a fair chance in the treatment group. And, you guys are all university students and I presume, have enough knowledge about biology and experimentation to know that, we can't ever evaluate the value of research, the value of a treatment unless we are able to compare it to something else. And so that's true also with these psychology experiments. If we don't do some of these psychology experiments, we don't know. So even like the most extreme cases, I mean, I don't think that you would do the prison study again or the shock study again, but until those studies were done, it wasn't really scientifically possible for us to predict that they would go so terribly, so there's even an argument to be made that you can do extreme things and you just have to have very specific concepts of when to cut them off. So there, again, going back to the coronavirus situation, what is the name of the hydroxychloroquine, right? So that's the favorite drug, the drug that the president claims that he was taking. There was a study that was cut short because it was found that it became harmful to a significant number of the patients, right? And you can debate legitimately about how harmful, et cetera, and whether, the other studies should have been, so other studies were ended that based on this line of experimentation. And so in retrospect, maybe we were giving patients something that was causing harm, but unless we did that experiment, we wouldn't have known about that.
I think those are all really valid points. I did want to add, though, I think something that I've been really concerned about recently, taking like global health and public health, is like the outsourcing of medicinal testing and how it's usually exported to countries where people are more at risk of not, or I guess are less likely to be able to pay, so they are more at risk of being exposed to the harmful effects of these medicinal studies. Like I think it was in Zimbabwe.
Yes.
Yes.
I was just thinking about that, and then you said it. Yeah.
They did all, like, they would cut off like, I think it was AIDS. I'm not fully sure at the moment, but yeah, like there have been multiple studies across like dozens of countries in the global south where people will go and they will test medicine and people will die, and they'll get like maybe like, like paltry like dollar amounts are in like the hundreds or the thousands, and they come back and the finished product is used, and like that's like kind of embedded in the system. So I really think we should consider how harmful it is, because we wouldn't have birth control if it wasn't tested on Puerto Rican women and like experimented on Black American women. So like the whole system, like.
So what's the solution? So this is an interesting question for you guys. I've been thinking about this a lot because there are literally thousands of people your age out on the street right now. And so we need to talk about solutions. So yes, there are these types of like experimental inequities, but how do we actually fix some of those systems? I mean, I don't know if that's what you want to talk about now because this is a journal club, but I'm really inspired that young people like you are thinking about these things. And but I encourage you to move in a direction of actionable recommendations, right? What do you really want to happen? It's great. It's important that we recognize the inequities, but what do we do about them?
Well, I think I was reading some more studies, all the studies in this file inspired me to read a few more studies. And I don't know if you guys have heard about like this study about rats, how if they're given like drugs, addictive drugs, they're much less likely to take them if they are in a stable environment and they have stimulation from like other rats that are also related to them. So I was thinking of, and I was also reading another study about how like, I think it was in Egypt, they had a holistic medicine. Like, I think we should move toward holistic medicine in a lot of different ways, as in, not as in, like, just like homeopathy, but as in like, we have to treat nutrition and like mental wellness as a part of physical wellness. And until we do that, the trauma responses that we have, especially linked to epigenetics, are going to be like extremely high. So, I mean, that's one thing, of course. But yeah, I don't think there's a direct solution unless we systematically overhaul a lot of things. But I don't know. I think it's important to ask questions.
Yeah, one thing I wanted to mention with Huma's point is that like it goes just beyond like science, because like some of those inequalities, like you see them in our solutions to like just global problems as well. So like one thing Huma mentioned was Zimbabwe, and a couple of years ago I remember Zimbabwe was going through a famine, and the UN solution to that was to give them like tons and tons of GMO food. But the problem with that was that like we don't know like the long-term effects of like eating GMO food for like that long, right? So that's kind of why Zimbabwe's government turned all that food down. It was just because their statement was like, why should our people be guinea pigs for something that the rest of the world has not even like tested or indulged in? And I think like a really important piece to like addressing these inequities and inequalities and stuff is that like all of our textbooks, everything that we learn in university, like it doesn't really like go over stuff like this, right? Because I feel like most of the stuff that like we learn either by seeing like a news article about it on our free time and stuff like that, and we kind of have to educate ourselves on that, and that kind of like just perpetuates the problem in the system, and the fact that like we're not taught these things even in our like science courses and stuff like that. So I feel like before we can begin at, like going about like potential solutions to like address it, a really important thing is to like just educate ourselves on it, and like yeah, just like educate and raise awareness about it so that we can see that it's a problem in the first place.
I do agree with you on that. I think the most important thing is not the trade of goods or money. It's more of the trade of ideas. Because if you give a country a lot of money, they can use it quickly. And then what are they going to do? They don't have any money anymore. But if you teach them how to create an economy or how to produce more money, then they will continuously improve that, and so on and so forth. So I think in this case, the trade of ideas is probably more important than the trades of goods and services. And in terms of the science part of using human subjects, there are also a lot of animal models that we have noticed that are good, that overlap with humans, rats, monkeys. Of course, we have to think about animal rights as well. But a really cool thing about science these days is that we have the technology to produce single cell lines. So if we're trying to focus on one specific cell line, we could just test the drug or the hormone on that single cell line without worrying about how an animal or a human feels during the experimentation.
One question I have about that with the single cell lines is, do you think that removing that cell from the like complex hormone and chemical interactions that it would experience within the body actually would like affect the outcomes of the study? Like what if in the single cell line the cell behaves in X way without certain hormonal chemical reactions, but when it's in an environment where those hormonal chemical reactions are present, the drug reacts differently within the cell, leading to like a harmful net effect rather than a beneficial net effect?
It could. The difference between single cell lines and having a more complex organism is that in a complex organism like a human or a rat, there's a lot more variables happening. So a single cell line would be better used for something a little more simple, like testing one single drug. But if you're trying to understand the rest of the body, it would be a lot harder to understand for a single cell line. But a good example of the use of a single cell line is growing viruses and making a vaccine, which is very important nowadays, of course, with COVID. So instead of growing the virus in like a human, which is very, very wrong, you should never do that. You can do it in a single cell line, like in this case, probably a bat cell line or something. So it would be more applicable to like maybe a single hormone virus growth, something like that. But of course, it does get a little more complicated when you have to include other variables like multiple hormones, or how one hormone affects more than one organ system, because obviously we don't have the technology to grow one huge organ system. That's a little too complicated right now.
Yeah, but I think that the single cell lines that Ruby's talking about, I think that a lot of pharmaceutical companies use them anyway to kind of get like a preliminary basis of like what effect the drug has on like a cellular level to begin with anyway. So that's a really good point.
Personally, I do research in neurochemistry on campus, obviously not on campus right now, but when I did do research in the neurochemistry campus, we did use chromaffin cells, which are very important in the exocytosis of like adrenaline and all those really important hormones that control stress. And we use those to understand the exocytosis of various molecules. We don't know what molecules they are yet, but it's very important. We did this because it was really important to understand opioid production and the production of opioid peptides in the body, which in turn would help understand like how the human body can produce opioids on its own instead of having to take opiates, which in that part contributes to the opioid crisis, which is a whole nother rabbit hole I do not want to get into.
I think all the ethical discussions have been really good from both sides. You know, there's always pros and cons to conducting an experiment and how ethical it is. It can be very difficult to fully like, you know, make something ethical or to get the best results if it's ethical. And that's like a really big discussion. Maybe someone can do a presentation on it. But I just wanted to quickly, before Dr. Hartstone presents, show you guys our blog, which would be another great way to talk about this. So I linked it, so it's in all the emails we send now. You can just click on the link and go straight to this blog, and it brings you to this page. And as you can see, these are the recent posts since all the club officers presented last time. You can see all of our posts are down here, so hopefully Zainab and Humza can put their posts in the next couple of days, and we can, you know, have a continued discussion about it. So you can click on anyone's post. Hassan had a really good he had a really good presentation last time, so if you want to go look at his slides, they're there, and then you can just leave a reply down here. And that way, if you weren't able to make it to a club meeting, or if someone you know wanted to learn about the club or isn't a state member, they can definitely just go on the blog and look at it. And then if you go to contact, we linked our YouTube and our Instagram as well. So we record all of these, and we post them on our YouTube. So you guys can actually go and listen to the presentations as well. If you look at the blog and you think someone's is interesting, just go watch their presentation. So that's it. So Dr. Hartstone can present now.
All right. So I guess before I start talking about these papers that I think you were all given access to, I'd like to know, who are you? Why are you here? What are you doing? So I learned, so at least one of you is not an NC State student, one of the presenters, yes?
Hi. Sir, right here. I attend the illustrious North Carolina Central University, so just had to put that out there.
That's
Eagle pride.
So this club is not just for state students, or are you like a special guest in it, or?
Yeah, no, I'm just kidding. I'm actually really good friends with the presidents, and they actually asked if I was free to present. So I was like, why would I missed out on that amazing opportunity.
Okay,
yeah.
that's cool. And why does this club exist? Why are you guys doing this?
So we founded this club because I know I was really bummed out when my summer internship got canceled. And then I realized that a lot of people were also in the same boat as me. And I really love research. I've been doing research every year that I've been at State. So I got together with my other pre-med friends, Rabia, Hassan, and Shabazz. And we decided to start this club so we could keep on having like productive conversations about research the way that we are right now. And I know it won't actually like simulate like being in the lab, of course, but at least we'll still be thinking, we'll still be reading about research. And I know that reading research is a really, really big part of actually conducting research in the lab. So I wanted all of us to like kind of develop the skill before the semester starts and before we go back into those positions in the lab and stuff like that.
Okay, and what kind of lab do you work in? Whose lab? Which lab?
I actually don't work on campus. I work at the NIH in Durham. So I've been working there since my sophomore year. So.
Oh, wow. And what year are you now?
I'm starting my senior year now. Yeah.
Okay, and most of you guys are pre-med?
I think all of us are. And I think Shabazz and Hassan may have done research on campus as well, if they want to mention what lab they were in.
Yeah, so for me, I was supposed to be doing BitSure this year, this biotech research with the biotech department, obviously, with Dr. Sengupta. Obviously, I can't be in lab, but like we're doing like online stuff more so. But like the STEM Journal Club, like Myra said, like it's part of like developing certain skills, whether it's like reading research articles or just being exposed to different science topics that I don't really know about.
Okay. And I guess you guys have like these little reaction things. You could like raise your hand and tell me if you are in a research lab or doing research with some kind of professor or organization. Got one thumbs up from Ruby.
Yeah, I do research on campus with Dr. Sombers, Leslie Sombers. In her neurochemistry lab, we do a lot of, it's more of an interdisciplinary lab between neuroscience and electrochemistry. Basically, my work that I do a lot is with electrodes and using that to analyze single cell exocytosis, specifically in chromaffin cells. So it's basically like preliminary, that's a big word. It's basically work to better understand the release of opioid peptides in the human body. So instead of giving patients opiates, we can just have the body produce more opioids, which correlates to how you feel pain in the body. So if you have the human body release more opioids instead of giving it opiates, you have a lesser chance of increasing addiction and such, and you can still relieve pain in very painful situations, I guess. Yeah.
Okay, that sounds pretty great. So it looks like most of you are not involved in research, though. You should be.
I think like for the majority of us, like including myself, I'm gonna be a sophomore at NC State next year, and like most of the research that over the summer positions, they got canceled. So any research that, like even like I was like trying to get into neuro research, but they emailed me and was like, oh yeah, we're canceled for the summer. So when Myra reached out and was like, hi, like this is the idea I have, like are you interested? I was like, yes, of course I'm interested. And I think the good part about this is like in the lab, like you learn about one specific research and that's like basically what you focus on. But with this club, you can really learn about like a lot of different things. Even today we saw that like it was a psychology experiment to like learning about like cancer.
Yeah.
So I think it's like very interesting because you get to learn about very like different aspects of science, and sometimes people have like a very like narrow view of what like research or science can be, but in reality it's like such a wide variety of topics.
Yeah. Well, I think that my main sort of emphasis for any of you guys, obviously you're all very like academically engaged. And so I think all of you should try to weasel your way into research labs. It's super important. You know, no matter what you want to do in life, assuming that college is not the end for you, if you want to go to graduate school or medical school or whatever, That's the kind of thing that's really going to make a difference. So I run a pretty big research lab. So I generally have a couple postdocs and some graduate students and the lab manager. And at any given time, I have usually about 24 undergrads that work in my lab. And we study functional morphology and comparative anatomy. So basically how animals are adapted to do what they do. So that's the main gist of our research. And we always, have any of you guys ever heard of my lab before, or any of you in the zoology club? I came and talked to the zoo club a little while ago. No. I always tell students when I'm trying to introduce them to our lab that we study functional morphology in terms of all of the levels of deadness. So from live animals, that's the least dead you can be. So we do like behavioral research, and then we do a lot of dissection. So those are freshly dead. So we do a lot of muscle research. And then more dead than that are bones and teeth. So we have in our collection about 700 skeletal specimens and skulls, and we go to museums all over the world to study bones and teeth. And then the most dead is fossils. So we do a lot of paleontology in my lab as well. And so the papers that I sent to you today are kind of along that spectrum. And I pulled them specifically because they are related to student research, which is what I wanted to encourage you. So remind me which papers. I sent you the hyoid paper that we just published. So the
Yeah,
direwolf one.
you
Yeah,
said the hyoid paper, the one about the pseudothumb,
yeah.
and the one about musteloids.
Musteloids, right. Okay, so those are 3 papers that are, well, so which one do you want to start with? Does anybody have a vote?
I read the Lemur paper. I liked it.
The Lemur paper is super cool. Let's start with that. Let me find it. Yeah. Okay, so I can pull it up. How do I do my share screen? The green button that says share screen. Look at that. Okay, there we go. All right, can you guys see this thing? So this guy here is, what kind of animal is this?
An eye lemur.
An eye lemur, yeah. So that's the correct answer. I also would have accepted the ugliest damn animal I've ever seen on Earth. So yeah, so the eye is a ridiculous looking animal. Does anybody know anything that is special about the eye besides obviously the pseudothumb, which we'll talk about?
They do have some, so the people that live near the eye, they are really gravely endangered because the people that live around it think that it's like a like symbol of death or something like that. So
can you blame them? It's a horrible looking thing.
So yeah, they're like ordered to kill them on site because they are a symbol of death. So,
yeah. So where do they live?
Madagascar, I believe.
Yeah. So Madagascar. So John Joyce thinks they're really cute. No, I think they're horrible. So yeah, so they live in Madagascar. They're a lemur. So lemurs only live in the island of Madagascar, which is like, that's a technical truth, but there are some other things that are basically lemurs that we don't call lemurs because they don't live in Madagascar. So like some of you might know about lorises or galagos or bush babies, which they're lemurish things, but since they're not in Madagascar, they're technically not lemurs. But the eye is a lemur. It's a very special lemur. So it is the largest nocturnal primate in the entire world. So most other nocturnal primates are much smaller than the eye. It is the only member of its entire family. So there are no other lemurs. So even though it's a lemur, it's the only kind of lemur that's remotely similar to itself. There's nothing that's very similar to it. Does anybody know what's special about its teeth? So eyes have ever-growing incisors. Their teeth grow continuously throughout their life. So that's more like a rodent than like you or me, right? So all other primates have like 2 sets of teeth, and that's the end of the story. And then they have these giant ears. You can't really tell from this picture because I kind of made it really dark like that, but they have huge ears and they have these crazy hands. And so if you see, I guess I can zoom in and try to get like focus on the hands. Oh, that's like really close to a creepy animal. So if you look at these crazy hands, they have a super long middle finger. So this third finger here is really long and really skinny. Here's the skeleton of the middle finger. Can you guys see my cursor as I move it on the screen? Yeah, okay, so there's the skeleton. And so this little skinny finger, they use this and those huge ears and those ever-growing incisors to do a crazy behavior called tap foraging, where they find a dead piece of wood and they take that skinny little middle finger and they tap it 7 or 8 times per second. So it's super fast, like almost unimaginably fast. They're tapping on the wood and they are actually using their giant ears to echolocate voids in the wood. So they find like grooves that were carved by grubs and other insects in the decaying wood. And then they take those ever-growing incisors and they bite into the wood and create a little hole into the tunnel, and then they stick that disgusting middle finger into the hole and fish out grubs. And so that's a substantial part of their diet. They eat the grubs there. And so we were dissecting some eyes and studying their forearms. I've also published a bunch of papers on their masticatory muscles. I'm not really doing a good job keeping up with the chats. Does any other mammal echolocate? Well, there's one big group of mammals that echolocates. What's the main type of mammals that echolocate?
I like whales.
Is it dolphins?
So whales do, yep. So that there are 2 other groups, right? So whales do echolocation, and then there's one other group that does echolocation. What's the other group that does echolocation? Bats, yeah. So bats and whales, and basically eyes. It's a really rare ability. Eyes are actually able to do an amazing thing. So with bats, they're hearing like the insects. Whales, you know, they're getting reverberations off of fish, etc. The eyes are actually creating a 3-dimensional map in their head of what's happening under the surface of the wood. And so when they bite a hole into the wood, it's not in a random spot. It's like it's right at an intersection of different pathways so that they have like maximum efficiency of being able to fish out the grubs. So really amazing. So anyway, so we were studying their head muscles and we were studying their forearm muscles. I published a few papers on eye forearms. When we were dissecting the eye, we actually found a crazy thing, which is, let me zoom out. So we were dissecting it and we noticed that there was this little structure in basically near the wrist of the eye, close to the base of the thumb. And what was super cool is that we found that there were 3 muscles that attached to it. So if you see this purple muscle here, can you guys still see my screen? Yeah. So this purple tendon here is a muscle called abductor pollicis longus, which sounds kind of crazy, but it's actually really simple. So abduction means to move away from the body. So the way I always teach my students is like when you abduct a small child, you're stealing it, you're taking it away, right? So abduction is moving it away. So abductor pollicis, the pollex is the thumb, and longus means that it's the long muscle because there's also a short one that's actually in the hands. Anyway, so this purple tendon, abductor pollicis longus, it usually attaches to the thumb itself, right? So here's the thumb, but it happens to send a little piece of the tendon over to this weird little structure. So there's this red bone and this pink little piece of cartilage. And those structures are what is called a prepollex, which basically means before the thumb. And not only does that muscle move that bone and cartilage, but there's another muscle, this yellow muscle, and then there's another muscle as well, the green bit. And so this little structure is moved by 3 different muscles. And so it's really like an anatomical system. And so one of the muscles pulls it out. So the abductor pollicis pulls it out. The opponens pollicis pulls it in, so it adducts it. And then there's this green one, which pulls it across towards the pinky side. And what was super cool, so we started looking at this at other different eyes. So we found this, we studied it in about 3 different eyes, or no, we looked at more than 3. We found that it's not in other lemurs. So these are these other lemur species. And what was amazing, when we started looking at it really closely, we discovered that the pseudo thumb actually has its own fingerprint, which is really cool. And so basically this is an accessory digit. It's an extra finger on an II, and it's the first primate that's ever been identified to have this extra finger. And what we think is going on is we think that the II basically, it evolved this ridiculous hand, like this crazy hand that's super good for the tap foraging, but it's terrible for grabbing on stuff, right? So like if you need to grab on stuff with any type of force, you want to have short stubby fingers. And so these long skinny fingers are not very good for gripping. And so we think that the II evolved this extra digit in order to compensate for how terrible its regular fingers are for grabbing onto stuff. And so that's what we found. And then there's this other kind of neat story that goes along with that, which is that this pseudo thumb, this prepollex, this other structure that exists in the II, it exists in some other mammals. And the most famous example is in the giant panda. So the panda famously has a pseudo thumb. So if you look at the hand of a panda, it has like a thumb and then it has other fingers. But if you count those other fingers, like those other claws, you'll realize that there are 5 fingers, not like 4 fingers and a thumb, but a thumb and then 5 other fingers. And so the panda evolved this exact same structure. And what's super cool is that it evolved the same bone and cartilage, and the 3 muscles that control it do the exact same thing in the giant panda's hand. And the reason for that is because pandas need to have the ability to grip bamboo. And so other bears, they lost their thumb. It basically became just another one of the fingers. And the reason that they lost their thumb is the same reason that we have such stupid big toes. Like we can't do anything good with our feet, right? Because our big toe became in line with all of our other little itty bitty toes. Whereas like our cool relatives like chimpanzees or this II is hanging from his feet. So he has way cooler feet than we do. But the problem is that when you're walking around, our big toes would be like flapping on each other and damaging each other. And so for efficiency of walking upright, we had to get rid of our big toes. And the same thing happened in bears. They got rid of their thumbs because their thumbs, they would have been stepping on themselves. And so what happened was that this one lineage of bear, they did something really stupid, which is they started eating bamboo, which is a terrible decision for a lot of reasons. It's a bad evolutionary trajectory. Bamboo is a horrible food. It's bad quality. That's why bamboo or pandas sit around all day long. They look really cute, but they're really just like lazy bears that are just processing everything. And like, eating massive quantities and pooping it all out and like, being really lame. And so, but in order to eat that bamboo, they had to be able to grab it. And so they needed to evolve a thumb again. And so they had 2 options, evolutionarily speaking. They could either split out that original thumb again, or what they ended up doing was evolving this structure in their wrist. So almost in the same way that the II did. So that was really cool. And it got a lot of attention. It was in the New York Times and National Geographic and Smithsonian and a whole bunch of newspapers all over the world. We released this paper just before Halloween because we were hoping that like this creepy animal would get like more attention, because it's like a living version of a gremlin. So what questions do you have about this research?
I had a question. I went to the Duke Lemur Center like kind of near the end of summer, and they're like basically most of the animals in Madagascar are like not endangered or like almost extinct.
Mm-hmm.
And so like, was it difficult doing research on lemurs? Like, would you consider it ethical, since like we had a lot of discussion about that?
Well, so that's an interesting question. So I have dissected hundreds of animals in my life. Hundreds. I have the heads of animals in my office, hundreds of them. But I've never once killed an animal for my research. It's pretty crazy. Yeah, so all of our animals, like you can't go and you can't kill an II. You could probably get away with it in Madagascar because it's not exactly like it's kind of a lawless country, let's say. But no, I would never do that. And so these animals all died. In fact, this animal that's being photographed here, and the main specimen from our study, is from the Duke Lemur Center. And of course, the Lemur Center would never let us kill anything. So this photograph is of an ii from the Lemur Center. This hand is from a Lemur Center ii as well. But yeah. So yeah, I don't think that there's any ethical issues. At NC State, you don't have to get ethical permission to work on dead animals. And so in fact, I feel like an ethical obligation to learn as much as possible about the anatomy of especially endangered animals. And so like these iis or these lemurs that live at Duke, or the iis that live at Duke, they are used for research during their lives. And when they die, I want to get as much out of them as possible in terms of knowledge that we can bring to the world. But that's a good question. I'm glad you asked it, because sometimes people leave talks with me thinking that like I'm just like a lemur masochist. So I'm glad you gave me the opportunity to dispel that myth.
So do you think you'll find like pseudo-thumbs in other animals? Are you going to pursue research in other animals?
That's a great question. So there are some other rodents that we know have them, like that needs a grasp, like stalks of grass and things like that. One of my students is looking at the hands of some marsupials that we think might have them. I'm really interested in bats, actually. So obviously, so what happens with the hand of a bat? Evolutionarily speaking, where is the hand of a bat?
The wings.
The wings, right. So it's really interesting. Like wings evolved a bunch of times in nature, right? So insects have wings, but just looking at vertebrates, wings evolved in bats. What else did wings evolve in?
Birds.
Birds, that's the other obvious one. Does anybody know the third really good vertebrate example of wing evolution? I'll give you a clue. It doesn't exist anymore.
Pterodactyls?
The pterodactyls, exactly. And so in these three vertebrate examples, the wings evolved very differently. So a bird wing is from basically the whole limb. So like if you eat like, I'm sorry if you can hear my dog freaking out in the background. It's really horrible to work from home these days. I'm really desperate to get back to my office. Anyway, so a bird wing evolved from the whole upper limb, right? So like when you're eating like chicken wings on like the Super Bowl, right? So the little drumstick looking thing, that's the humerus. And then what they call the flats, the two bone part, that's the radius and ulna. And then the little itty bitty dinky part they usually cut off, and they don't serve you that during the Super Bowl. But that's the whole upper limb, right? Whereas pterosaurs, their whole wing, they also have most of the upper wing, but most of their wing is one finger, right? So it's one long finger. In bats, their wing is mostly hand, right? And so they have these giant hands. The problem is that a lot of bats have to like go and walk around on stuff. So they might climb on things. And some bats evolved like little flippery things like right at the crook of their wrist. And so we think that they might have something akin to the pseudo-thumb. So we're about to study that as well. All right. Do you have more II questions, or do you want to talk about some of these other ones?
I was wondering about the clicking, because I know that like people can also do that. What do you think that this research can potentially mean for human biology?
That's a really good question. That's a really, really good question. I get asked a lot about the significance. So I often feel guilty, like during like this coronavirus pandemic. I'm not sure that this has any meaning to humanity, other than that it's kind of fundamentally cool to learn something about one of our creepy relatives. So I really consider myself like a pure scientist. So like any of you who have contributed to like even the smallest little bit of cancer research, like you have probably made a bigger impact on like humanity than I have. My entire impact on humanity is inspiring students and teaching students to do scientific inquisition. And so I think that if IIs went extinct, like it would be a tragic loss, but I think we would survive. I don't think that it has a lot of implications for like human echolocation. I'm a little bit skeptical about the human echolocation. I've seen like one blind guy is pretty impressive and can do like some amazing things, but I don't exactly think that that's like a main trajectory that humanity is about to take.
That's pretty valid. I did learn about limb, like how people develop bones depending, like people also develop different not necessarily appendages, but like smaller bones that can like develop based on what we do. And we, like, younger kids are like growing up without, there's like a tendon in your hand, and also their thumbs are different because of how we use it. Do you think, I mean, I thought this was really interesting because of that reason. Like, I would like to know, like, how, like, do you think that people could develop something similar? Obviously not like another limb, but like
Oh yeah, that'd be
more
cool. Like if
muscle.
we could like text faster, if we had 4 thumbs instead of just 2, maybe
Yeah.
maybe that's the direction that humanity is going. I mean, it's possible. So one of the things that I think is interesting about this is that the exact same complex evolved between aye-ayes and bears. And in fact, the rodents that have the same structures, it's literally like the same 3 muscles and the bone and the cartilage. And what that means is that this anatomy is within all of us, right? And so what I think happens is that it's really simple stuff. Like if a gene stays on a little bit longer, then you just keep adding fingers. And if you turn that gene off a little bit, then you reduce fingers. And that's the difference between like a primate and a horse, right? A horse, it turned that gene off earlier during development. And so it just walks around on one toe. And there are functional reasons that that's useful. So animals that are that do a lot of running, they want to have fewer fingers, like cows and horses. Animals that do a lot of grasping, they want to have more fingers and toes. That's why humans and lemurs have all of these things. But I think that the differences there are really subtle little controls. And so maybe, I mean, if you want to pioneer like an industry of like raising a generation of children that are better at sending text messages, then maybe you can leave on that like extra thumb gene for a while and we can see where that takes us. It's an interesting idea. That's not an endorsement, but it might be within all of us. All right, let me quickly talk about these other 2 papers that we were talking about. So one was the musteloid paper. That is this one. Can you see this paper now?
Yes.
Okay. And then the other one is the hyoid paper. Let me first talk about the hyoid paper. So that's Flores. So now you can see the hyoid paper? Yes? No?
This is the musteloid paper.
The musteloid paper? Okay, I'm going to stop for a second and then I'm going to share again. You'd think that by now I would know much better about how to actually use Zoom, but I'm still pretty crappy at it. Okay, so this is a paper that just came out recently. In fact, it's still in what's called the online version, so it's not in the fully like on paper version. And it was written by one of my former students. So Deanna graduated from my lab last May, so May of 2019. Emma is still in my lab. She's a rising senior. Aaron, oh no, I think she's a rising junior, and Erin is a rising senior. And then Edwin is my postdoc. And so this is a really neat paper. So what this is a paper about is about the throat bones of dogs, of canids from Rancho La Brea. Have you guys ever heard of Rancho La Brea? The Rancho La Brea tar pits? No? Okay, let me go back to. So the tar pits, wait, I can't see all of you, but has anybody seen Rancho La Brea? Maybe in the chats? Nobody. Okay, so Rancho La Brea, if any of you have been to Los Angeles and you did not go to Rancho La Brea, then you have tragically missed out on the coolest fossil locality in the entire world, in my opinion. So what it is what's called a tar seep. So tar is literally bubbling out of the ground in a park in the middle of Los Angeles. It's crazy. Let that sink in for a second. So have any of you been to Los Angeles or that part of like Southern California? Yeah. So do you remember seeing oil wells? Like there are oil rigs like in the middle of town that are like pumping up crude oil, which I thought was really,
they're like all over the place over there.
They're all over the place because there's all of this crude. And so tar is basically like a form of crude oil. I don't know if that's literally true, but just go with me on it. I'm a biologist, not a chemist. But and so what's amazing is, so there's a park right in the middle of Los Angeles off Wilshire Boulevard. In fact, the park is there because the tar is there, and you really can't like build a building on top of it, otherwise it would like sink and be unstable and all of that. And what's neat is that that this tar has been seeping out of the ground for tens of thousands of years since the last ice age, and tens of thousands of years before the end of the last ice age. And what happens is that this tar seeps up and it forms kind of little ponds. Sometimes it's just like a little mound. Sometimes it's basically like a lake. And when it rains in Southern California, which is very rare, but when it rains, water collects on the top of these like tar lakes. And this again has been happening for thousands, tens of thousands of years. And animals would come from far and wide, and they would think that they could go and like get a drink of water there. And so they go up to the edge of the water and they drink it, and I'm sure it tastes nasty because it's like tar, right? And they would get their feet stuck in the tar, and they'd start to sink down, and then they'd start to struggle, and carnivores hurt them. And there were amazing carnivores. So the herbivores were things like mammoths and giant ground sloths and cool things like that. And at that time, the place was full of saber-toothed tigers and dire wolves. So two of like the coolest ice age carnivores. And they would hear these herbivores struggling, and they would go and they'd think that they could get like an easy snack. And so they'd go and they'd probably eat on that like animal for a while. And then when they'd jump off, they would also get stuck in the tar, and everything just started sinking down, and it formed layer upon layer of the most pristine bones. So what it literally is, so the goo is called tar, but we call the whole fossiliferic amalgamation, we call it asphalt. So it's just like asphalt on the street. So asphalt on the street is tar that is mixed with little stones, right? That's what like highways are made out of. But instead of the stones, all of that stuff is the bones. So all of these different bones. And so you have perfect skulls and skeletons of all of these really cool species that are perfectly preserved. And they're preserved so well that we can literally pull out the littlest bones from the specimen. So my other students, Erin, in fact, she's doing her senior thesis studying the ossicles, the ear bones of all of these different carnivores from Rancho La Brea. So you can pull out, about ear bones, the three smallest bones in your body are the malleus, incus, and stapes. And so they're literally just millimeters long. And so they're perfectly preserved. And so what Rancho La Brea, and there are thousands of them. So there are over, I think, 3,000 dire wolves that have been identified from La Brea, and like over 1,000 saber-toothed tigers and giant bears and things like that. And all of the bones are so beautiful that we can actually study, like for the first time, samples that we can study statistically. And also we can look at bones that are generally understudied in paleontology. And so this paper is the third paper that I have published on what we call the rare bones of Rancho La Brea. So the first one that we published was on the clavicles of Rancho La Brea, so the collarbones of saber-toothed tigers and this giant cat called the American lion. And then the next paper we published was really exciting. I should have sent you this paper. That was a paper on the penis bones from Rancho La Brea. So you guys might not know this, but a lot of mammals have bones in their genitals, and that's especially true for carnivores. And so we studied the penis bones of dire wolves, and that paper got a lot of attention. And my favorite conference presentation that I ever gave was called "Extraordinary Penises: The Bacula of Rancho La Brea." So that was very, very popular. There's a whole podcast that my scientific conclusion was about how size does matter for dire wolves. And then, so this is the third one where we studied the throat bones. And what was really cool is, so we took all of these measurements. So here you can see all the measurements that Deanna took. So in humans, we have one hyoid bone. And really our hyoid is a fusion of a few of these bones. And the rest of these are basically like ligaments in humans. But in carnivores, they have either 7 or 9 of these throat bones. And so Deanna studied them, and her conclusion was that dire wolves have a really different shaped hyoid complex. So you can see that the dire wolves are significantly different in shape from gray wolves, and she also compared them to red wolves and to coyotes. And what she basically figured out, and she thinks that dire wolves, from this we can almost infer that dire wolves maybe had a deeper voice than modern wolves. And so they howled at a lower frequency. And so now she wrote another paper where we're talking about she thinks that saber-toothed tigers were capable of roaring instead of purring. They could roar. And so that was really exciting. And so the reason that I wanted you guys to read this is because this is like a legitimate piece of research that was conducted by someone at your stage of career. And yes, it took like a year for her to publish it because the publication process is long. But this is a project that my one student basically led, and other undergraduates helped her collect those data. And she, not only that, but she got like funding. So the three of them got grants to go out to California to collect these data. And they came with me. We got other funding to go to the Smithsonian to look at comparative specimens. And so it was super fun. And then the musteloid paper that we presented, that was a paper that another one of my students really worked on, and that was a dissection paper. Okay, questions about this, or about, I was hoping that we could have like maybe conversations about like undergraduate research. I think that might be useful for you. What would you like to, oh, look at this guy. So everybody, Drew Patel just joined, if you see. He's kind of snuck in here quietly. Do any of you guys know Drew?
Yeah, I'm really good friends with Drew. And when you were talking about your lab and how you have dozens of students working for you, I remember that Drew told me that he worked in a lab and the name sounded like with all these animal models and stuff like that. So I remembered him. So I texted him and I was like, oh, I think like, what lab do you work with? And he said Hartstone. And I was like, oh, he's talking to us right now. You should join. And then he came.
Yeah. So there's Drew. He's one of the greats. And then Elisa Schwenk also joined. She's another one of my lab students. So I asked them, like, I hope that's okay, but I shared the URL for the Zoom. So I asked them if they wanted to come so that we can talk about what it's like to do research. And I don't have to tell Drew this, but he knows that if he can talk crap about being in lab, and he often does. So he'll tell you for real what it's like.
I've said it before. I just don't like the writing part of it. Everything else we do in the lab is really cool, especially like the
writing.
The writing is not my favorite part.
Some students really hate the writing, and other students are good at it. Some are good at it and enjoy it. Some are good at it and hate it. Drew just hates it. Elisa is really good at writing, actually, but I don't think she enjoys it at all. No.
I hate writing.
You hate writing? But Elisa, how many papers have you published in my lab now? Just one, the II paper?
Yeah.
Aren't you on the Floyd paper also, or no?
No, that's Rudy.
Yeah. So both Drew and Elisa are working on different research than me. So they're mostly working on what we call our digital dissection stuff. So Drew is doing this cool project where he's scanning the skulls, like using different surface scanning techniques of different carnivores and correlating that to different types of data that we have, like the musteloid paper. So correlating like the shape of the skull to like the bite forces and things like that from the musteloids. And then Elisa is doing more digital dissection stuff. So we've got these scans of different lemur skulls, for instance, and we're dissecting their muscles in 3-dimensional space. It's really amazing.
I actually had a quick question, if you don't mind me asking.
Of course, please.
So I guess like kind of getting to start that research and kind of like finding a research topic, and I guess those resources that people can help you. Like, how would you recommend getting started at that?
So there's one overarching thing that you must do if you want to do research as an undergrad, in my opinion. Like the number one thing that you should do to start, and that is you have to find a mentor. So weasel your way into a lab, right? And so what happens in my lab, and Drew's a great example of this because he's been in my lab for a long time actually now.
Yeah, since freshman year.
Yeah, and so he's done a little bit of pretty much everything in the lab, right? I mean, you helped with dissections, you've helped with like, you came to the museum to do work with us. Like he's done a lot. And then over the years, he kind of figured out like what his main role was in the lab and like what he became interested in and started developing questions. So at the beginning, like my students kind of work on either questions that I've generated or other students' questions. And then by the time they become seniors, you know, and more senior in the lab, like Drew, then they are able to sort of generate their own direction. But it all starts with, you have to find a mentor who's studying something that's of interest to you. So Drew, he wants to go to dental school. So, you know, at first he thought he was going to be interested in like doing kind of the tooth research in my lab, and he's gotten into like skulls. And so, and it relates to mastication. So it's like it makes sense for his dental school applications. Elisa wants to go into medicine. So she wants to go to medical school. And so she was interested in some of the dissection based work, but she's really like in more of the 21st century space where she's working on like that digital space, which is super cool. But yeah, it all starts with, they had to like weasel their way into my lab. And from there, they've learned, you know, how to do research.
Do you have any suggestions?
Okay, cool. Thank you.
Yeah. Who was talking? Was that Hassan?
Yeah, that was me. Do you have any suggestions on how someone would weasel their way into a lab?
Drew, why don't you talk about that? You're pretty weaselly.
Pretty weaselly. Well, I didn't have to, because we had talked originally when you had first moved here from South Carolina when you had started. So I just go ahead.
No, I mean, but so like you met me basically. So all of you have met professors. Hopefully you've introduced yourself to your professors at like the front of the room. If you haven't, then you should definitely make a point to do that. But meet your professors. You can go on their websites, or you can go at NC State. We have what we call the Curious website. I can't remember what Curious stands for. It stands for something, but there are different professors that will like advertise positions on there. But the number one thing I think is meet professors. And if you think that they're interesting, then talk to them, find out if they have a research lab, or if they don't, then they can recommend like somebody to work with. So is Dr. Galler like the mentor for this club? Is that why he was on at the beginning?
Yes, sir.
Yeah. He's a mentor for this club. Yeah. And
yeah,
so I think he was a mentor for the BitShare program as well. And the program. So that's how I got into touch with him. And so like for me, for like everyone here who's like interested in research, for me it was really hard. Like I was just shy like about talking to a professor one-on-one. But one thing I did my freshman year is that the NC State website, they have a whole list of like research programs that you can apply to for the summer. So I applied and I got into the Be More program my freshman year summer. And that kind of is what broke me into research. And then from there, I developed a really strong relationship with Dr. Galler, my mentor. And from there, I could transition into doing like research during the semester too.
Yeah, so that was going to be my main recommendation. So Dr. Galler is like the sweetest guy, and he knows like the biology department really well. And so if you're interested in research, then talk to him about like your general interests or what you want to get out of a mentorship. So you might not have like a realm of research, but you might want like a mentor that's going to like work very closely with you, or work like you might want one that gives you more autonomy, or, you know, like talk to him about those kind of things, and he can think through like all of the different options that we have in the department.
Yeah.
Dr. Hartstone, I remember you mentioned, I like visited your lab once. You mentioned that there was a class that you have to take prior to doing research in your lab.
So we don't have any prereqs for doing research in my lab, but the way that I have managed to teach so many students is that I've basically taken like my approach to mentorship and turned it into a class. And so all of the students that want to do research in my class, they basically sign up for a 3-credit class. And actually we have 2 classes. So we have like a fall class and a spring class. And the idea is like, my recommendation is students should always like plan on being in my lab for at least a year. Because if you're in my lab for less than a year, then you're not really going to get like the full experience. Or honestly, like I'm sure that as Elisa or Drew will tell you, like you don't really know what the hell's going on until like at least the second year. And so all of the students, so they end up getting credit for that. And then after they have basically credited out, right? So Drew's been in my lab so long, he couldn't possibly keep taking classes with me. And so is Elisa now. They become kind of mentors themselves, and they help teach. So like each of them gave like lectures to my students, and they mentor my new students. So next semester I have about maybe 18 or 20 students that are staying on, including Drew and Elisa. And then I've got 13 new students that are joining. And so Elisa and Drew will help them get up to speed and learn that stuff. And like you were saying, like you could be shy and sometimes people are intimidated. I think I'm a very approachable person, but like I think Elisa will tell you that she was scared of me for like a long time. And so it makes it easier for like the new students to like, you know, be able to check in with like the older students for mentorship as well.
And the class itself is also split up. So like the first semester you do is more about writing, which I didn't like that part as much, but it's where you learn how to write, you read some papers, how to analyze the papers and stuff. And then the second semester, you learn research methods like dissection, maceration, actual using JMP.
Yeah, so different types of software, different types of statistics, like we use a lot of statistics.
Yep.
The other reason that I like to have my students take this as a class is because, in my opinion, this is the most important thing that you could do as an undergrad. I know that you have to get your major requirements done and you've got all of these other classes that you need to take, but what's going to set you apart from all of the other people who are applying to medical school or dental school or vet school is the rich experience. This is what you're going to write about in your like applications, you know, and when you're being interviewed. And so a lot of people think of research as like an extra thing that the students can do or like you can do in your spare time. And I wanted to make sure that my students were able to carve out at least 6 credits in their schedule to like focus on this. And I think that my main students, like my students that really stick with it, like Drew and Lisa, they would tell you that this is like the place that they hang out. Like they hang out in my lab and they spend way more time in my lab than they would, than they are just for credit, for instance. Sometimes we feel like we can't get rid of Drew. He's like a plague of some sort. But a little bit. No, we're always joking that he's secretly in charge of lab. What other questions?
For students that don't go to state, but like associate universities or colleges, would you still would you recommend kind of like the same stuff? Because I feel like for me, it's a little bit easier to reach out to people or professors who go to my school, but it's like the scope of the research isn't quite like what I'm interested in. Like it's a little bit harder to reach out to professors at different colleges. Do you have any advice for that?
Yeah, that's a really good point. Where do you go to school? Anyone? Okay.
Sorry, I just got muted. I go to school at Meredith.
Oh, at Meredith. That's crazy. You're a current Meredith student?
Yes, I am.
What year are you?
I'm a rising senior.
Oh, that's great. So my PhD student who just graduated is going to become a biology professor. She's just got a job at Meredith. And so she's going to be like a mentor that Meredith students could join the lab. And she's obviously going to do similar stuff to what we do. So NC State is what we call an R1 institution. Have you guys ever heard of that terminology? A Carnegie R1 institution. And that means that research is actually the primary mission of the university. So obviously, like, we are an educational institution, but the tenure track professors, the core faculty, are dedicated, like, for instance, people like me, as much time as I spend with the students, technically my job is always at a minimum 51% research, and like 30% teaching, and then there's service. In fact, I think right now I'm supposed to be like 70% research. And so institutions like us, research is our main goal. Institutions like Meredith, research is a minority goal. So like at Meredith, the professors are 90% supposed to be focused on teaching. And so obviously at institutions like that, and then you have other places like Western Carolina, it's going to be more equal between research and teaching, stuff like that. So there's different types of institutions. And so I recommend reaching out to professors. So at Meredith, you can reach out to NC State professors. You can just come down the street, but there are people who are doing research at all different types of institutions. They might be doing it more over the summer than over the school year. But I think that the key is just reach out, just, bother people, and sometimes it'll stick, The worst thing that happens, like, They just might not reply to your email, which I think is really rude. I'll at least reply to your email. And we have trouble turning people away. So we always have a whole bunch of students, and unfortunately we do have to turn people down. But you know, most professors I think will take a student or two, or you know, a small handful of students. But it's up to you. I mean, I think the research experience is more important for you than it is for us to have you. So it's on you to reach out. You have to do this.
Huma, one thing I would also say as well is that in my experience, NC State summer programs, I think they reserve a certain number of spots just for state students. And in the program I did, we were only allowed 2 NC State students in that program. And then the other 6, 7 spots were for other students. So if you have time for next year, make sure you apply to those NC State summer programs, because that could be a really great way for you to get research. So.
And also, Dr. Goller has a summer program himself, and so he knows a lot about the summer program stuff as well. So he's a great person to talk to about that. He'll help you figure that out too.
Yeah, thank you both. Yeah, I did actually apply to a lot of those, but I think most of them got canceled. But,
yeah,
so
I mean, this summer, like I was supposed to be giving a presentation in Greece like this week, and then I was taking a whole bunch of students to Paris to do research in July next month, and that all got canceled. It's horrible.
Also, I know, like right now, I was supposed to have an internship at the NHI, and they're doing webinars, and it's pretty much like it's for the people in the program, but it's also for like basically anyone else also. So if you want the links to them, like it's kind of how to do research, how to like have resilience, how to like all that type of stuff. Like they're doing seminars about it, and they're also posting them. So if you want that, I can email that out after.
Yeah, that's nice. But I think the key is we have to get through this stupid pandemic, and then you should like try to really get into a lab and do stuff in the lab.
If you send us those links, we can send them out to everyone if you want to.
Of course.
Personally, I just emailed like 30 plus professors, and I just bothered Dr. Sombers a lot, and she finally let me in.
Yeah.
So,
be annoying.
Emailing people.
I mean, there's kind of a method. So mostly it's that professors are busy. So like, you know, and honestly, I get like probably something on the order of 100 emails a day. So sometimes emails get buried. So there's like something to the persistence. But also, if you're annoying, it shows that you've got like follow through, and that's what we want too. So, you know, don't be shy. The key is, you know, just try to muscle your way in. What else?
Do you think that, like, with what happened this summer, if you go into the school you're looking for research, do you think that like a lack of research experience will ever stop you from getting into a lab? Like if you're an upperclassman but you haven't had any research experience, labs will turn you down? Or?
So that's a really good question. So first of all, lack of research experience has never hurt a student applying to my lab. I prefer to take students that know absolutely nothing. And I think, no offense, but I think professors basically always assume that you don't know anything anyway. So like, have you noticed that anytime you take a class, even like if you've taken the prereq, they still cover all of that same material again in the first few weeks? Like, you know, we basically assume that you're coming in from the ground zero. So that I think will never hurt you in an application. What will hurt you though is that I think, and this is something that you guys should do. So you guys are mostly upperclassmen, and that means that you have an obligation to mentor younger classmen as well. Get people involved early. So try to convince people to do research. Don't wait until your junior or senior year to try to organize this. If you are already a junior or senior, then, you know, don't let me discourage you. Like, go out and try to find that opportunity and push your way in. But definitely tell like all your younger siblings, your sorority mates or fraternity mates, or like any younger people that especially involved in clubs like this. Freshman and sophomore year, I think, is like kind of the ideal time to try to weasel your way into a space, especially because, like I said, students don't really understand anything that's going on in my lab until they've been there for like a whole year. So yeah, but if you're a senior, it's not too late.
Something that I also wanted to add, So for me, I transferred after my sophomore year.
Mhm.
So I transferred to state after my sophomore year. So like when I came in, like, you know, like Dr. Hartstone said, like most of the time they're looking freshmen, sophomore year, but then like juniors and seniors, they're like, I don't know. But for me, like I made sure to emphasize like, hey, like I just transferred though.
Yeah.
And
That's really important. So, transfer students are kind of like a special case. In fact, like I think of you guys as like the hidden, not minority, but we have to take special care of transfer students because you have that shorter amount of time on campus. Oftentimes your schedules are fuller because you have to get like all of those required classes under your belt, and so you're at like a systematic disadvantage, which I think is really scary and dangerous and is a disservice to you. And so I always pay very close attention to transfer students. And in fact, my lab manager is a transfer student, and she's an absolutely wonderful person and a great science researcher. But we have to be very cognizant that you have to get up to speed. You're trying to cram in, you know, a whole R1 experience, right? So NC State's the R1 school. You're trying to cram in all of that R1 experience in just like the last 2 or 3 years. So it's not so easy.
Right, for sure. And some advice, like from me at least. So I'm doing BitShore this summer, and like obviously like I have to do it at home, but like regardless, like I still like got in and stuff like that. And for me, it had a lot to do with the fact that my mentor for BitShore was a professor I took a class with last semester.
Yeah.
So like my professor told me straight up, like, if you didn't take the class with me last semester, you probably wouldn't have even gotten an interview.
Oh man.
So
Well,
like,
yeah, so build a relationship with those professors. Again, build a relationship with Carlos, who is like such a sweet guy, and I know will be a champion for all of you as part of this club. You know, if you're interested in the kinds of things I was talking to you about, you're welcome to email me and we can talk about this stuff. But it's on you. You all have to build that group of professors that can be your kind of support system. And you're all trying to go places, right? You want to go to medical school or wherever. So you're going to need those recommendations as well. Drew, you looked like you had something you wanted to say.
No, but I can say something if you want me to.
I don't know. Elisa, do you have any other advice for them? No. Elisa is shy, but she's super sweet that she came to this.
No, I'm mean.
She's mean. She always says she's mean.
Yeah, the only thing is for me, like, as you said, I'm pretty shy, so I didn't want to join the lab at first because I was really afraid that I wasn't going to, like, know anything and I was going to be, like, embarrassing myself. But you just kind of have to put yourself out there. I still embarrass myself anyway, and I still don't know what I'm doing, but it's okay. Everyone else, like, no one else knows what they're doing either. Like, you're not the only one. It's fine.
Yeah, I mean, science is fundamentally like the experience of learning, right? We're just trying to like learn subject material. If you're doing really good science, like all of my students are studying something that we don't know what the answer is. So, you know, I've done more research, obviously, but I don't know the answer to the question that Drew's trying to ask. I don't know the answer to Elisa's question. So they can legitimately like make that discovery and become the expert themselves. So yeah, we're all clueless at some level. Drew's just more clueless than almost anybody I know, but
a little bit.
All right. Does anybody have anything else? This has been like I think this club's pretty cool. If you guys ever have trouble getting, you know, somebody else to come talk, I can convince like some of my other friends to do it. Or my wife is a psychology professor. Some of you might have taken class with her. So if you want to talk to somebody who's more qualified to talk about the prison study and stuff like that, you could try to organize that sometime.
Yes, we would love to have you back.
Yeah. Well, I'm happy to come back sometime. Just send me an email.
Yeah, for sure. Thank you so much for coming and talking to us about your research and giving us advice as well.
Yeah, you're welcome. I think you guys are an impressive bunch of people. It's nice to have such like a welcoming, supportive group. It's very cool that you have people from other institutions participating. I think you should try to build that. Also, I appreciate Drew and Elisa. Thank you for joining this too.
Yeah, thank you guys for coming. And Dr. Hartstone, if you want to recommend your other colleagues to come through, we're okay with that.
All right. What you need to do you guys have a t-shirt yet? So that's what I like. The zoology club, they always have really sharp t-shirts. So develop a t-shirt, shoot one into my mailbox, and I'll hook you up with some more speakers. So.
I got you.
Some STEM
Nobody can say no
journal
to a free
club
t-shirt.
swag. Yeah. All right, y'all. It's nice talking with you.
All right, thank you.
Yeah, bye.
Thank you.